This protocol is the light and dark bottle method from AP Environmental Science Unit on primary productivity, the same lab my classroom is running this week. Two bottles per condition. Same water, same plant mass, same light, same bottles. One stays clear, the other gets stored in the dark, cutting off photosynthesis completely. The dark bottle can only lose oxygen. The light bottle does both jobs at once. Subtract what the dark bottle lost from what the light bottle gained, and the difference is net primary productivity oxygen that is actually available to everything else in the water. If you would like to run this experiment for yourself with your own probe or DO test kit, you can find the instructions pinned in the top comment below. Also, apologies for some of the jumpy footage. This was all done for posterity's sake and for reproducibility should you wish to do the experiment yourself. Elodea, the plant the word oxygenator was basically built around, and appears in almost every biology photosynthesis lab you could find. Hornwort, sold under the same label, completely rootless. Filamentous algae, the wildcard entrant. Not a vascular plant, but we threw it in anyway. Rotala, an aquascaping favorite, and as far as the literature is concerned, essentially untested for oxygen output. And rounding us out is Java moss. No roots, no vascular tissue at all, sold to nearly every beginner in the hobby. Half of that lineup has research behind it, half of it does not. Elodea's photosynthesis has been measured directly against filamentous algae. A 1986 study running Elodea canadensis alongside two algae species found its own oxygen output holds it back more than either algae's does. Under the exact conditions a well-lit planted tank builds by afternoon, aka high pH and low CO2, that same study found the algae held its performance better than the Elodea did. Hornwort also carries the oxygenator label commercially as a plant and is often sold under it. In 2018, it was tested side by side with Elodea. Hornwort held higher dissolved oxygen over time. The other plant's own density ended up working against it. Enough biomass in one space to shade itself and consume more oxygen than it produced. A separate study, run for a NASA funded life support research program, measured hornwort's peak rate directly, about 75 nanomoles of oxygen per gram of dry weight per second under optimal CO2 conditions. Java Moss doesn't have a comparative number attached to it, but it shows up by name in a real DO probe teaching protocol nearly identical to the one you will see here. A well known, well behaved oxygen producing organism, just never measured competitively. Rotala turns up constantly in aquascaping guides. They turn up nowhere in the photosynthesis literature. A search for comparative number turns nothing rigorous. The plants people actually buy now are the ones nobody's actually measured. There's a second variable I decided to run through every bottle where the water came from. Tap water starts close to a blank slate, treated, no established biology of its own. Aquarium water is already a running system with bacteria, biofilm, dissolved organics inside. A background respiration rate that has nothing to do with whatever plant gets added. Which is why this design needs a control for each water source on its own. A bottle with no plant in it at all, run through the same light and dark cycle to measure what the water was already doing before a single leaf went in. Skip that step, and every reading gets credited to a plant that didn't necessarily earn it. Based on what's published, here's what should happen. Elodea and hornwort would likely lead, especially in tap water where there is less buffering capacity to keep the pH from climbing. Algae has no obvious reason to underperform under those same conditions, though the water source should not matter as much. Java moss is likely the floor here, not necessarily from being a poor performer, but because mosses as a group run slower metabolisms than plants with actual vascular tissue. Rotala is the real test. There's no published number to check against. It's also the only one that is solely rooted. That's the honest answer. The hypothesis of the guy who teaches this subject and runs this lab every year. Algae's aquarium water bottle finished at 13 milligrams of oxygen per liter, almost double where it started, in a sealed bottle. Corrected against the control, that's 0.20 milligrams per liter per hour, the highest number in the entire lineup. Switched to tap water, and Algae's number fell by nearly two-thirds, down to 0.07. Elodea barely moved, 0.19 to 0.16. The only species that stayed roughly consistent across both the aquarium and tap water sources. Whatever aquarium water was giving to the algae, tap water wasn't giving it back. Rotala, a species with no published number to check against, finished last in both water sources. In tap water, its corrected NPP was 0.01, barely distinguishable from a bottle with no plant in it at all. The predicted floor was Java moss, however, it wasn't. This moss beat the plant literally marked as an oxygenator on both tap water measurements. One number complicates the respiration side of this. Every plant's dark bottle read 0.8 milligrams per liter after 48 hours. 5 different species, one identical number. That's a floor, not a coincidence. And it means the GPP figures here are probably underestimates of the true rate. Trust the NPP numbers over the GPP ones from this run. NPP doesn't depend on the dark bottle at all. One prediction did hold exactly. Both controls lost oxygen even in the light, and the aquarium water control lost more than the tap water one. Precisely the background activity gap we predicted. A version of this exact math was taught in this room this week on a curriculum that never asks which specific plant does the job best. 5 species, 2 water sources. Now there's a number for each one. Enough to make one wonder about other species. We'll revisit this again, this time next year, with even more species. I hope you'll join us then.